Good afternoon, everyone. Welcome to this workshop, Mathematics of Cryo-EM. And actually, I've seen a lot of people from life science side and some people from mathematics side. Of course, in the future, as a mathematician, I actually want to see more people from mathematics and computer science side to make this a truly interdisciplinary research area, a strength of HKUST. So this afternoon, actually, before I came here, I also I had a meeting with our Vice President of Research and Development, Nancy Yip. So Professor Yip asked me to also welcome you on her behalf. Okay. And I want to maybe just say a couple of things. Oh, by the way, my name is Yang Wang. I'm the Dean of Science here. So I've been hearing enough from BIC about, well, let's do what Tsinghua has been doing, okay? So, they got 3, Cryo-EM machines, and, this is a very exciting area. Actually, BIC has, I've seen your work with, collaboration with Tsinghua. I was actually very impressed by the, at least, from the picture, I was blown away, actually. Yeah. So, since then, we've been hearing, me and Nancy, our vice president, and also our provost have been hearing quite a lot about the exciting breakthroughs in the area, the kind of things that allow us to do, and that we certainly would like to have an active research area in this area as well. So we've been working together, and I must say, really the effort has been mostly led by BIC. We're really also thinking of having a machine here and building up a research team in this area. So this workshop is actually very timely because for research in this area to thrive, we not only need top experimentalists and but also the strength in computation. Okay, so I think this workshop from the title alone, I think it will bring people in these 2 areas together, maybe even more areas. Okay, so you have come here, many of you. Some of you probably, like Amit, I talked to him this morning. He came here for the first time and really enjoyed the campus here. I hope for our guests here, you will have a great time enjoying our campus. The weather seems to be okay, but please do leave before Sunday because I think there is a typhoon coming in, okay? Anyway, I just want to give my welcome here and thanks for coming. Thanks for all of you for coming here.
Okay, so before I introduce my buddy collaborator, Gao Ning, I want to just add a plug for how mathematicians can help contribute to life science. Since Watson and Crick in 1962, when they did their breakthrough work on solving the structure of double helix, of course they won a Nobel Prize. Since then, 33 Nobel Prizes have been awarded to structures in biological molecules or approaches towards imaging structures. So for biologists, for life scientists, seeing the structure is a big part of what we do. So this is why this half-day symposium is timely because in the past, all those structures have been solved mainly from X-ray crystallography. And since 2013, there's a breakthrough in Cryo-EM technology that allows us to look at very large molecules that are not crystallizable, and we can resolve them down to near atomic range. So with that, just to plug in for the mathematicians, if you help us, because, I forgot to mention, the reason why there's this big advancement in Cryo-EM, besides the hardware, better electron detectors, is the improved or new algorithm for reconstructing these 2D images into 3D structures. So this is where the mathematics comes in, and this is where you can contribute to important problems in life science. So with that, I would like to introduce Professor Ning Gao from Peking University, where he has been there for a few months. And he moved across the road from Tsinghua University, where he was assistant, associate, and then full professor and chair professor. That's where he was. So before that, he got his PhD with the important authority in Cryo-EM, Jakob Frank in Wadsworth Center in Albany. And then he was a postdoctoral associate with him at Columbia University before coming back to China. So in other words, Professor Gao was in Cryo-EM before this craze, this fad. So he really came back in the right time and so on. And just show you so we are collaborators, and we've been collaborators since 2013. That's the year that people call year zero of the resolution revolution. So from our collaboration in 2015, we published our first paper, and at that time, 2015, it was only 2 years ago, okay? At that time, he was an untenured assistant professor in Tsinghua University, and now he's full professor, chair professor at Peking University. So in the 2 years of this meteoric rise in rank, you can see that this is part of the growth of Cryo-EM. So here's Professor Gao.
First of all, I would like to thank Peking for inviting me here. So it's really a pleasure to be here again, and I think in the same room, and to introduce the technique and the fields I'm right now working on. So, and also, I'm very grateful for this opportunity, because I myself would have a chance to meet all different mathematicians, so they might be able to give us very good advice in data processing, and also the future of the development of Cryo-EM. Because at the beginning, so I thought this talk is going to be focused on the basic introduction of Cryo-EM, because I know in the audience there will be a lot of the mathematicians and the people from different fields, different background. And I was told today that there are also going to be a lot of students from the Division of Life Sciences. So this is probably going to be a very good education material. So basically, I will give a little bit history about Cryo-EM and its development, and also why Cryo-EM has been a hot word for many scientists, especially the recent breakthroughs in the last 3 or 4 years. And then I will discuss the current limitations in Cryo-EM, including from the sample freezing to the data analysis. And probably, I think that in the talk afterward, and Professor Amit will probably introduce his solutions to these problems. So speaking of Cryo-EM, so we know that the data we use for 3D reconstruction are from the transmission electron microscope. So here is actually the first design of the electron microscope. And the right is the first commercial electron microscope made by the Siemens. So this invention of this machine was done nearly 100 years ago. It's in the 1930s. So the scientist Ernest Rutherford has been awarded the Nobel Prize for making the electron microscope. But actually, shortly after the invention of this microscope, scientists already start to apply this machine to study biological materials. this is actually one of the oldest paper I can find. So this is basically a one-page Nature paper published in 1934. So you can read from the title, Electron Microscopy of Biological Objects. So this paper actually reports two figures. One figure is literally the first EM picture of a biological object. So in this blow-up, you will see it's actually a picture of a plant leaf at 450 times magnification. Of course, at the time, we cannot get any detailed information from the picture. But it's important because it's the first experimental realization of the EM application on biological material. Why it's hard is because biological materials are very fragile. Under the radiation of electrons, it will be quickly destroyed. So in the following years, scientists has been improved the application of Cryo-EM on biological materials from both sides. One side is from the instrumentation side. The other side is that we can treat biological materials to prepare the sample to provide more tolerance for electron damage. One of the example is the scientist George Plady. He was at Rockefeller. So he actually was one of the first to apply to invent different staining technique. And also he made a modification to the differential centrifugation technique and combined both to electron microscopy. So one of his major contributions to the cell biology is that by improving the negative staining technique, he actually discovered there are many small particles inside the cell, which was later named as a ribosome. So after the invention of the first electron microscope in the following 20 to 30 years, actually cell biologists has used the microscope to discover many new features of the cell. So they have discovered the membrane system inside the cell, discovered the organelles and large particles within the cell, such as ribosome. Of course, we know that if we look at the first original paper by Plady, it was in the 1950s. So around the same time, an important contribution in the structural biology field is that people start to use X-ray crystallography to study the structures of proteins or other biological molecules. So then, of course, it's very natural for people to think if we can use electron microscope to study the structure of biological materials. So this is actually start to form the new idea of so-called electron crystallography. So the only difference is that instead of using X-ray as a light source, so then we are using electron microscope to collect the diffraction data. So the best thing about electron microscope is that it can work in two modes. So we can set it in image mode, then collect data for phase information. And we can switch to diffraction mode for collecting data for amplitude information. And we'll combine both information to calculate the high-resolution structure. So what is the basic advantage of electrons over X-ray in crystallography is that X-ray usually has a very low scattering ability with the biological materials, but electron will interact stronger with biological material. So basically, for get useful information in X-ray crystallography, we need to grow the crystal into a very large size. But for if you are using electron microscope for collecting diffraction data, we don't need the crystal in that size. Usually, it can be nanometer-sized crystals, or even we can look at a single molecule with multiple with highly symmetric subunits within the molecule. So one example is actually the first 3D structure solved from the data collected using EM. So this paper was published in 1986. So basically, it's a reconstruction of the three-dimensional structure of the phage tail. So the phage tail is a helical structure. This is original structure. This is how the diffraction pattern looks like. And this is finally the reconstruction structure. So the pioneer of the electron crystallography is Aaron Klug, who is at the MRC. So he's awarded Nobel Prize in Chemistry in 1982 for his contribution in development of electron crystallography. Of course, in a helical molecule, we will have multiple repeats of the same subunit. So the same strategy can be applied to other highly symmetry molecules, such as virus. So in fact, the first 3D structure of a virus was also solved using electron crystallography. So so this paper was published in 1970. So Aaron Klug, again, with his student, reported the first structure of the virus. Later on, people realized if we can allow proteins, particularly membrane proteins, to form 2D crystals, then we can put these 2D crystals inside an electron microscope, then collect the data, and use the same electron crystallography to solve the structure of a membrane protein. So what you are looking at is the first 3D structure of a membrane protein. So although in the initial paper, the resolution was very low, only at 9 angstrom resolution, only enough to see the transmembrane helices. But this is actually the first structure of the membrane protein. And actually in 1980s, people were thinking that the future of Cryo-EM is 2D crystallography. So many people actually working on the crystallization of membrane proteins. They trying to make membrane proteins to form 2D crystals. Now you have seen a few examples. All of them are with either crystals or highly symmetric molecules. But how about the object without symmetry? So Joachim Frank, who is also my PhD advisor, he is one of the first who first had the concept of single particle method. So if we look at the particles, for example, there are two forms of biological objects. So one form, for example, what you are looking at is a mitochondrion. So if we isolate mitochondria from a cell, we'll find there is no two identical mitochondria. So each one is different from the others. So that means for these samples, we cannot find two exactly the same mitochondria. But for other large biological molecules, if we can isolate multi-copies from a single cell, and then we spread them onto the EM grid and take a picture of them, then we will see in the field of view, we will see multiple images of a same complex. So Joachim Frank was one of the first who coined the term single particle. So what single particle means is that actually well separated, that means we don't want them to stay together, and unattached and structurally homogeneous particles, such as the virus, ribosome, and other large biological molecules. Then in a single image we collected, we will have multiple multiple projections. So they are actually produced from different 3D objects. But in principle, we can assume all these 3D objects, they are chemically and structurally or the same. So that means all the 2D projections can be regarded as a 2D projection of the single 3D object, but from different view angles. So then the task of getting a 3D structure is that if we can find the mathematical relationship between those 2D projections, then we can use computer algorithms to generate the original or to recover the original 3D density function in order to get a 3D structure. So this is pretty much the concept of 3D construction and the birth of a single particle method. But why we want to freeze the biological material in a very thin layer of ice? So why we need to do the electron microscopy at cryo condition? The reason is that actually scientists, when they are publishing the first paper, they realized that the radiation damage to biological samples is very important. So one solution to this is if we can cool down the biological samples, then we can minimize the radiation damage. Another good thing about the cryo condition is that then in a cryo condition, the sample will be freezed in near native state. So that means we can get the structure which can represent their native structure. So then next, a few, actually 3 scientists, they made a very important contribution to the Cryo-EM. So the first 2 is Bob Glaser at Berkeley and Container and Florida State. They were the first to bring the to use liquid nitrogen for data collection. So in the presence of liquid nitrogen, then the high frequency data could be better preserved. Then Swiss scientist Dobichel, he actually invented a device which can freeze the biological sample in a very thin layer of ice. Then I will introduce the device later. So when the sample was kept under very low temperature, then we can get better images, and the structure of the sample is much better preserved. So if we collect the data using the Cryo-EM, then we will have a better chance to get higher resolution structures. So this short movie actually is a very nice introduction of the workflow of Cryo-EM. So you will see in the Embeded tube, there will be thousands of molecules. And this is we call the EM grid. So which basically is a sample support we use to deposit sample on the grid. And this machine is called Vitrobot. This is a machine we use to freeze the sample. So as you can see here, we only use very tiny. So the grid with a very thin layer of ice, thin layer of liquid will be quickly frozen. And during data collection, the grid will be kept in the liquid nitrogen temperature. Then we will take pictures of this ice embedded sample, and So this is one of the typical Cryo-EM images we collected on Cryo-EM. So you will see that the molecules are oriented randomly. Then we will use computer program to pick out those particles, and then we can do particle alignment and classification. Then mathematically, we can combine those 2D projections into a 3D structure. Depends on the data quality and the number of the particles or the number of the images. We can then determine the 3D structure and the different resolution. For example, what you are looking at probably around 10 angstrom resolution. At 10 angstrom, we start to see the secondary structure of a protein, such as alpha helix. So basically, the best thing about Cryo-EM is that it is a very flexible tool. So it can start all kinds of biological samples. For example, with so-called single particles that can be either symmetric or asymmetric. And we can also use EM to study the structures of 2D and tubular crystals, and also 3D crystals, 3D microcrystals. And for inside cell, there are many filament and fibers made of proteins. We can also use Cryo-EM to study those helical structures. Most importantly, in cell biology field, people have to use Cryo-EM to study the 3D structure of the whole cell or tissue sections. And the so-called electron tomography will be very important to the cell biology. But Cryo-EM only gets very famous in the recent 3 years. This is because there's two major breakthroughs. One major breakthrough, just as Peking mentioned, is a new detector. So the so-called direct electron detector is a new generation of detector. Here are 3 vendors. And another major breakthrough has been emerging algorithm for initial model calculation and efficient 2D and 3D classification. For example, I have mentioned here the Relan and the Crystallography, and 2 of the most popular softwares used by Cryo-EM groups. And our next speaker, Amit Singer, his lab also developed another Cryo-EM software. It's called Aspire. I think he will probably introduce some features of Aspire. So then I will give you a very short explanation why those breakthroughs has fundamentally transformed the Cryo-EM. So what is the electron direct detector? This is that the traditional way of collecting data on EM is we use a CCD camera. So there will be 2 times of singular conversion. First, electron to photons. Then the photons will be converted to electron again. Then we will store the singular in a digital image. But for the new detector, there is no such singular conversion. We will have materials that directly sensitive to electrons and then can count the electrons. So in this way, we don't have those singular conversions. Then the signal can be much better preserved. Also, in the new generation of the camera, the chip can be made very thin. That means the singular spread is less in the new generation of camera. So the good thing of the new camera is that it is very sensitive. So it can detect a single electron event. Then we can use a very low dose for image. That means if we detect a single electron event, a labeling, for example, when an electron hits this physical pixel, the neighboring pixels will also have a response. And then based on the response value of those neighboring pixels, then we can actually determine the position of this electron at sub-pixel accuracy. So this is called super resolution. Another good thing about the new camera is that it's very sensitive and it's very fast. And then instead of collecting a single frame image, such as for example, when we are using the CCD camera, sometimes we need to collect data for the total exposure time of 2 seconds. So for 2 seconds, we only get one image. But the new detector is very fast. Then instead of collecting a single frame image, we are actually collecting a movie. For example, for the 2 seconds of exposure, at most we can get nearly 1,000 frames. So the good thing about the movie mode is that then we can do the so-called motion correction. So what is motion correction? It's that we know that although the sample, those particles were freezed in a thin layer of ice, but once the electron beam hits the sample, those particles will move. So they not only move to one direction, actually they will rotate in the ice layer. So here, if you look at this picture, this shows the movement between individual frames. So the scale is actually angstrom. So if we look at those two dots, the movement is very big. So it can be as large, for example, from here to here, it's almost 20 angstrom. So that means in a traditional way of imaging, collecting data, for example, the CCD camera, in the 2-second exposure time, so the particle has moved from one place to another place. So that means in the actual image, we already lost our high-resolution data. So with all this benefit of the new generation of camera, so as I said, another breakthrough is a new generation of algorithm for 2-dimensional and 3-dimensional classification. That means, as I said, in our perception, the 3D reconstruction is that for all the images we have collected, they can be regarded as 2D projections of a same object. But in fact, biological materials or molecules, they are oscillating between different functional states in the solution. For example, in a set of data, except the major population, we often have other minor populations. So if we can do 3D classification, that means if we can separate the particles from different groups and then combine them into different groups and then perform 3D reconstruction within a group, we might be able to get multiple structures from a single data set. So that's the good thing about the 3D classification. This is also before the new generation of camera, we also perform the 3D classification, but we never get very good results. So I think this is a combined effect from both the algorithm development and the new device. So with all these two major breakthroughs in the past 3 years, there have been a burst of the important structures. For example, what you are looking at all the structure published before, after 2015 and before 2016. So you will see here there are the battery is out. You will see there's a very important viral structure, and down here you will see this is a splicing, and this is an important singular molecule, AAA1. It's a membrane protein. And here is basically a structure from our collaboration with Peking. So in the published work, the best resolution one can get from Cryo-EM images is 1.8 angstrom map. So now you can see at the 1.8 angstrom resolution, we can easily tell the difference of different amino acids. Then, for example, what you are looking at the 20 different amino acids and the model agreement, the agreement between the model and the actual density map. Then, next, I will introduce a few major challenges and limitations in Cryo-EM. But of course, mostly is from our experience. And the first is that the sample preparation. Then the next, I will introduce the current workflow of the data processing and the problems in the current workflow. And there will another big issue is that the modeling part, we will also need a new generation of software which can do the automatically modeling from Cryo-EM into actual models. So as I said, the device used for freezing sample was invented 30 years ago, but till now we are still using it. So the device is very simple. So basically we will apply a 3 to 5 microliter of sample on the EM grid, and then we use a pair, as you see in the movie, a pair of filter paper to blot most of the liquid away, and only the very thin layer of liquid on this grid. Then the grid is quickly frozen in the liquid icing. So afterwards, the grid will be kept in liquid nitrogen temperature all the way. So although there have been commercially available machines for doing this, for example, the FEI Witchbot and the EMGP from Leica, but all of them has a problem in many different ways. So before that, I will probably first introduce why this method, we really need someone to invent a new method. So what is a EM grid? So basically the grid is made from metal. So we use a supported sample. So it can be from different metals or differently sized. So normally its standard size is 3.5 millimeters in diameter. So basically we'll put another additional layer of material on the grid to support samples. So if we take a look at an EM grid under microscope, for example, here is a grid, then we will see there are many squares. So if we take a look at the one square, because we have covered this grid with a layer of carbon, and a layer of carbon film. In the carbon film, we have many holes, as you can see here. So if we further blow up one hole, you will see that we hope that a very thin layer of water will form a liquid suspension, and then your molecule of interest will stay in this suspension during the freezing. So if everything is okay, so then we will have a very good thin layer of ice in this hole. Then we take images at those holes. Yeah, for example, I mean, when everything works very good, so it's just like this. For example, what you are seeing is that it's a very good grid, and then we can take thousands of images of this. For example, what you are looking at is actually a ribosome images. So you will see that in a single micrograph, we will have probably hundreds of particles. And those particles are dispersed very well, and the image contrast is also very good. Then, as you can see here, we only use 17 hours for data collection. And if everything works as good as we expected, we might be able to get atomic structure in about a week. What I mean by atomic structure, what you can see here, and we can see the side chains of proteins very well. But this is not always the case. Actually, most of the times we will struggle with sample preparation. That means that sometimes we get a problem of we have to play with the ice thickness. For example, if the ice are too thin, so that means the sample will collapse. So what you are seeing here is that a big particle collapses into a random shape, and then although it looks like dark here, but we cannot get a meaningful 3D structure from those images. And often we will get a contamination of different things. For example, what you are seeing here are actually ice crystals during the sample preparation. So what a clean grid should look like this. And often we get sample aggregation. So samples, for certain samples, they tend to stay in clusters, or they will stay in the edge of a hole instead of in the middle of a hole. So this could be very painful because it will first waste most of your sample. Then you cannot find a good area for taking pictures. And in other cases, if we want to study a structure of a complex between several different components, although the major components is okay, but once you are adding new component, then you will get aggregation. So the problem is that the current device we use for a freezing sample is very hard to control, and it brings a lot of troubles. And another big issue is that we have a very serious problem for the preferred orientation. So that means certain biological molecules, they tend to stay in the ice in a single view. For example, what you are looking at is mostly top views. For example, if we collect all the particles, we will see that 93% of these particles are from these views. So if there is a problem of preferred orientation, usually we cannot get very good 3D structure from it. So that means we will need a new generation of a cryo-sample preparation device. It will use less sample, and it can give us a better distribution of particles. Most importantly, it should be able to provide good reproducibility, and then we can use this device to test the different conditions. There has been different attempts to introduce new elements into the sample preparation. So one such attempt is that instead of using a pair of filter paper to blot most of the liquid away, we can actually use a Microsprayer to spray a very tiny little droplet of samples on the EM grid, then quickly freeze the EM grid. So this is actually a very recent implementation by Joachim Frank and a group from engineering department at Columbia University. So in which they actually designed a Microsprayer. They used the microfluidics to make a chip. So in the chip, there will be the two input inlet. So one for solution A, the other for solution B. Then there will be an additional chamber to mix the two solutions. Then after the chamber, the sample will be quickly sprayed to the grid by nitrogen gas. So this device has two potential use. One is that the sample can be sprayed to the grid, and then it will require less sample. Another is that because there is a mixture of a chamber and there are fibers with the electron signal which can control the on and off of the two input, then we can potentially use this device for time-resolved sample preparation. So that means we can mix solution A and solution B for a time of 1 second, and for another batch of sample preparation, we can mix them for 3 seconds, and so on. So that's for sample preparation. I hope that there are some people from the engineering department which then can understand our problem. Probably someone will be interested in design a new device doing that. A big problem and a big challenge in the data processing is that, as I said, in the CCD camera, we only has one image. So basically, the computation need is not that big. But in the new generation camera, for each image, we'll have 30, 40, sometimes even have hundreds of frames. That means even for a single image, the data is huge. Then if we want to align the individual frames between in a single movie stack, then we need to have a very efficient algorithm to do that. For example, what you look at 4 different biological samples. So if we map the direction and the amplitude of movement for each individual particles, you will see that the particles, they move in different directions. So in different areas. That means in a traditional way of processing the movie images, we will need a better, efficient way. Probably we need to cut those big images into different smaller images and use those smaller area for alignment. Another big issue for that is that, as I said, during the data collection, the sample will be destroyed by electron beam. So that means in a movie data, for example, in a movie data, there will be many frames. So the damage to the individual frames are different. So for example, what you are looking at here is that here is a B factor of individual frames. So the B factor means that the motion of the particles in the individual frames. So you will see at the first several frames, the B factor are very large. That means when the electrons first hit the sample, it will induce very intensive movement. Then after a few frames, it will remain relatively constant. Then it will become larger again. So that means we can then analyze the particle movement between different frames, and then using this data to summarize, to make a averaged particle from different frames, but we will introduce another weighting function. That means each frame contributes differently to the final image. And also this weighting is dependent on frequency, because the radiation damage will first destroy the the high resolution features, then the low resolution features will be preserved much better. So the movie processing and the motion correction are very important for 3D structure. For example, before motion correction, we only get a map like this. But once the particles are properly processed, then we have a much improved map. And you can also see other cases. For example, if you look at the region of the nucleic acid, we will see that after the motion correction, using the new particles from motion correction, we will see the map is better resolved. Then we can see a separation, very good separation of bases. So the motion correction is important for getting high resolution structures, especially for smaller sized particles. Why for smaller sized particles? Because for large sized particles such as ribosome, the molecular weight is about 2.5 megadalton. Then it has many fine features. Then that means during the data analysis, we will have the alignment and the 3D classification can be performed at much higher accuracy. But for smaller sized particles, the contribution of singular from the individual pixels are much less. So that means the errors will be much larger. One extreme sample is like this. For example, what you are looking at is a very small membrane protein. It's around 300 kilodalton. It's actually 1/10 the size of the ribosome. Especially, it's totally embedded in a detergent molecule. So what you are looking at the 2D average of the images collected under similar condition. For this kind of particle, you will see no high resolution features on those 2D images. That means the alignment accuracy is not good. So why big particles tend to align very well is that, as I said, there are many fine features for alignment. But in this case, the singular is masked by non-specifically bound detergent. And also because the size of the molecule is much smaller, so that means the contribution from the singular contribution is much smaller. So we will need new tools in movie processing. That means what is movie processing is that for a single image, we will have 30, 40, or even 100 movie frames. So usually, if it's a 30-frame movie stack, we will have a file size of 7 gigabytes. It's huge, so it will cause a lot of computation burden. And we need a better statistical or mathematical models for dose-dependent frame alignment, and also for 3D classification. Before the introduction of the new camera, there have been many methods being introduced in the Cryo-EM field. Many of these methods, they perform very well on phantom data, but not so well on the real data. One reason is that the Cryo-EM data the singular to noise ratio is very, very low. So we need some new robust algorithm for processing the low singular to noise ratio data. So one example is that we probably need a new generation of software for particle picking. So what you are looking at is that after data collection, we will have hundreds and sometimes tens of hundreds images like this. Then we need to pick out particles for the further process. But this could be very easy if your quality of the sample is very good, if the contrast and the size of your sample is the size is very large and the quality is good, then it can be very easy for you to pick out. But for some of the samples, you will need a very efficient software to do that. Various methods has been used. For example, the pattern recognition, temporal-based searching, and machine learning. But none of them works on, in general sense, works on all different situations. And there is a lot of room for improvement. Then after particle picking, we will use 2D and 3D classification to get 3D structures. So one application of 3D classification is that we can group particles into different groups, then perform 3D reconstruction within these groups. Then we can get different structures. For example, what you're looking at actually 4 different structures from a single data set. So what I mean, 4 different structures, not just the structures are different and their compositions also vary a little bit. For example, in structure 1, you will see more proteins. In other structure, you will see less proteins. So this reflects that in solution, a big complex tends to have some certain component dissociating from the big one. So that means one purpose of 3D classifications is that we can find different subpopulations. Another purpose for 3D classification is that, for example, what you are looking at here is that we use 3D classification to calculate a few 3D structures and only include the best one for further 3D refinement. So for this purpose, we will throw most of the data away and only use a very small fraction of the data for high-resolution refinement. So that means to pick out the most homogeneous subset for high-resolution refinement. Although there has been a progress of softwares in doing this, for example, the ReLang, but there are many difficult situations. So we definitely need mathematicians to come in and to generate new tools for us. For example, we probably need an iterative particle picking program. I mean, based on information from initial 3D reconstruction. Then it can read the structure information from the 3D model and go back at the beginning to pick particles again. Then we will need a very easy method for particle sorting. For example, after the particle picking, usually there will be millions of particles. And in many cases, and nearly half of them are just garbage. But the graduate student tends to keep all of them because they always feel, okay, I don't want to throw a single good particle. So we might, if there is a very efficient way to quickly, to evaluate the quality of the particles, we can safely throw some of them. Then we'll decrease the computation burden afterwards. Then in the case of preferred orientation, so-called preferred orientation is that, for example, in a data set, we'll have 90% of particle stay in the same wheel. So then we will perform a so-called deep 2D classification. Because for data like that, most of the 2D class average will be almost the same. So that means we probably need a better way to classify particles into different groups. Especially, we will need an efficient way to find the minor populations within the sample. That means it's very easy to find a major population. But as I said, for many of the Cryo-EM project, we actually threw out 70%, 80% of our particles away. There are many important information in those thorough data set. So we need to have a way to find those important information. So in the future, I think that there could be a lot of opportunities for both instrumentation and the software side. For the instrumentation side, so one is probably the faceplate, and didn't have time to touch on that point. And another is that, as I mentioned, we need a high throughput method for sample freezing. So this probably we need a new concept of freezing instead of using the old device. For the software, we probably need a robust automatic data collection. So that means make data collection much easier to free the hands from electron microscope operation. And we need better software for particle picking, screening, and also flexible algorithm for classification, especially for the samples with very serious heterogeneity. And one important issue is that, as I said, though the images are very big in size, so we need a very efficient method for handling image frames. So I think that the more breakthroughs are actually forthcoming with the participation of experts from different fields of science. So actually, in the history, all the major breakthroughs of Cryo-EM are not from biologists. They are from physicists or from engineering, from all different areas of science. So with that said, I would like to thank the grant agencies in China. So my research was funded mostly by Ministry of Science and Technology and also by the National Electoral Science Foundation of China. And I have a lot of collaborators. And here is a picture of my group the picture was taken at Tsinghua. So the members are the same. So also I would like to thank the Cryo-EM facility at Tsinghua, especially the director of the Tsinghua facility, Dr. Jenny Lei. So thank you very much.
I have a question from machine learning maybe. When you mentioned particle picking, is that easy to get labeled data to just judge whether the window contains a particle or not?
What do you mean by labeled data?
In machine learning, people use ImageNet to ask whether the image contains certain object like a cat or a dog. Whether that's natural object, very easy to judge for common people. They just go to the internet, use crowdsourcing to put a label. But for this very specific image, is that easy to get such label data?
We will rely on manual picking probably for like 10% of the data. We will firstly manually pick a few particles based on user knowledge, and then this information can be used by the program.
So the 10% may be very high quality, means clear windows with object.
But in the end, we would like to have one, just as you said, it's a fully automatic one, so without any user intervention. I mean, currently, we rely largely on user knowledge and user experience in particle picking.
So what is the smallest protein the Cryo-EM can comfortably work with right now?
Comfortably work with, I would say probably 100 to 150 kilodalton. But there are already in literature that people get I think 6 or 7 angstrom structure from 60 kilodalton protein. Okay. So that, I mean that in principle we can work with very small proteins.
Is there any examples of using Cryo-EM to sort structure of RNAs?
RNAs?
Yeah.
Yes.
There is.
Yeah, for example, the structure of the ribosome, because ribosome, there are many ribosome RNAs. Yeah.
That's RNA, I mean.
RNA alone, you mean?
Yeah.
Because RNA alone used to be very flexible. So there are other cases that if you can get a stable structure of the RNA-P, yes. So if you know your binding partner of a certain RNA, so then you can use Cryo-EM to solve that structure.
Yeah, I have two questions. First, the 3D structure can be predicted based on primary sequence, right? So how does this two field merge, your Cryo-EM and the prediction, computer-based prediction field? Will these two data go together somehow?
Yeah, I think at the last step, the modeling step.
Yeah.
So usually, after we have a 3D density map, we need to translate the 3D density map into a model, into an actual protein structure model. So normally we will use the 3D structure prediction data and to find roughly the position of those predicted model inside the 3D density map, and using this as a start model to refine the structure.
Yeah, was that possible for the 70% of those image data you threw away can be integrated or somehow refined using this kind of modeling data?
Yeah.
Okay, second question is in the biology system, when you show the first EM of leaf tissue, right? So we use a microtome to cut the tissue in the slicing. Is that possible to cut the live cell into thin layer? You don't have to do the grid. You use that solution to suspend. You can look at the entire cell.
Yes, actually, in tomography, people will
reduce the tomography all the way to or expand to the cell level, rather than at single molecule level. Is that possible to achieve?
I mean, one problem is that the cell is very large, right? The cells are very large, so that's why we need to cut it into very thin sections, because the diameter of a cell is usually several micrometers. So if you put the whole cell in a microscope, you will see nothing, because the electrons cannot get through because it's too thick. So that means we need to have some way to get it thinner. So one thing is, as you said, to section it into different layers. But as you said, without sectioning,
if you can achieve very thin layer, then you don't need to do grid. You can look at those molecules just from a cutting machine.
Yes, actually, one particular area of tomography is that they use the so-called subtomogram averaging technique. So as I said here, so we are actually averaging individual particles at a two-dimensional level, and in a very thin layer of cell contents, there will have be thousands of single particles. Then we can get a 3D structure of a very thin layer of cellular content. Then there will be multiple copies of a same 3D object. When we can average them at the 3D structures level, then that can also be improved into near atomic resolution. For example, one best resolution one can get is around 4 angstrom.
When talking about the limitations, you have mentioned one of the limitations is about the preferred orientation. So my question is whether the preferred orientation is the nature of the particle or because of the buffers. Because if it is because of the buffers, then we can do something, change the condition to solve this problem, right? I'm just curious about that. Thank you.
Yeah. I think it should be the nature of the particle. So that means that the surface charge are not uniform. So and also the hydrophobicity are not uniform. So that's why they tend to stay or attached on the surface of grid, use only one side. In that case, we need to try different conditions. We change as you said, the buffer and the pH value, and change the salt concentration. Sometimes we need to treat the grid differently to increase the hydrophobicity of the grid, or sometimes we need to coat the grid with different materials. But in our case, we cannot predict which way will work. So basically, we have to try different solutions.
Okay, if there are no more questions, or if there are still more questions, I think we will first thank Professor Gao for his really illuminating talk.